Thank you for joining us. This is our final concurrent session of the day, part 3 in a series of sessions about water treatment tools and techniques. And we have 3 different speakers from our land grant institutions in the state of North Carolina to talk about solutions to different challenges that we have related to water treatment. Our first.
Good afternoon. My name is Jonaé Wood, and this is my project on the co-treatment of the food processing wastewater with municipal treated wastewater, along with my PI, Dr. WRRI. And like mentioned, I'm a master's student at North Carolina A&T State University. So a little background on our project. So the food processing industry is a major contributor of environmental pollution. The high levels of BOD5, COD, TSS, dissolved solids, and nitrogenous waste makes food processing water extremely costly and difficult to treat. On the other hand, municipal wastewater is also high in nitrates, even after secondary treatment. So our project seeks to combine both of these waters in a co-treatment process as a tertiary effort of remediating the environmental pollution and the risk. In this co-treatment process of wastewater, we have the food processing wastewater in which the carbon present will act as a reducing agent and an electron donor to convert carbonic products to carbon dioxide. Whereas in the municipal wastewater, we have nitrates being converted to nitrogen gas in the reduction process. And we can see this in our redox tower. And we see how both of these reactions will take place as well as their millivoltage value. Our research, we seek to answer 2 questions. The first question is, what would be the ideal ratio of both of these wastewaters to get the optimal pollutant removal? And how does the retention time affect the optimal removal rate using that ratio that we found in the previous question? So the objective of this research is to evaluate the most effective retention time and mixing ratio for the optimal treatment of food processing wastewater to municipal treated wastewater under slightly reducing conditions. Our wastewater was sourced locally from the water reclamation facility in Greensboro for our municipal treated wastewater, and the North Carolina Food Innovation Lab in Kannapolis, where they were producing legumes, pea extract to be exact. We also sought to stimulate both wastewaters, and we made some of them synthetically as well. So to do this project, we intended to use a sequencing batch reactor, which has been proven to be successful in treating wastewater by the food processing industry. And this reaction has 5 steps, which is the filling step, is number 1 that takes place for 33% of the time. The reaction step takes place also for 33% of the time. And then we have the settling phase, the drawing phase, and the idle phase. And we withdrew water from the system 2 times. The first time was after the filling step at the end of stage 1, and the second was at the end of stage 4, the drawing stage, which is when the water should have been treated after that second time. So in this experiment, we have 5 treatments that are replicated 3 times with ratios representing 0, 25, 50, 75, and 100% of food processing wastewater. And our food processing to municipal wastewater ratios were 0 to 1, 1 to 3, 1 to 1, 3 to 1, and 1 to 0. The entire reaction took place over the length of 600 minutes. And this pump here, I think it's the wrong one. This pump here is what we use for aeration. And like I mentioned, sampling occurs at the end of the filling step and the drawing stage. So in 98 minutes into our experiment, as well as 574 minutes into our experiment, we sample the water each time. So this is our experimental setup. We use glass bottles with the ability to withdraw water from them. We also have rubber stoppers that has pipettes in them with tubes connected to our air pump so that we are allowed to aerate our experiment. And these stages, like I mentioned twice before, is where we sample our water. So in this first stage, the filling stage, the water has yet to be treated. And in this second sampling stage, the drawing stage, we should have our treated wastewater. So for our analysis, we use trial codes that told us how our wastewater was made. So we have SS representing the stimulated. We have SR representing the stimulated and real. SSS, where we used a bit of seeded municipal wastewater, about 5 mL of municipal wastewater to 1,500 of synthetic wastewater. And then we have RR representing the real from both wastewater sources. We also have the equation for enhanced removal of both carbon and nitrogen, in which we took the actual concentration from the concentration expected by dilution, and we divided by the actual concentration. And to get the percentage, we times that by, multiplied that by 100. So the first thing that we got was our pH. And from our pH data, we can tell that real wastewater stays towards neutral. And the treatment process has the ability to bring those wastewaters back towards neutral, which is it's not too acidic and it's not too basic. And it doesn't deviate very much from being around the neutral area too. So in the ORP table, we can see that just as we expected our ORP values to be for the conversion processes, we have those same ORP values expected, which is around 500 to 700 for the carbon, and around, sorry, I may be mixing that up, but 500 and around as low as around 50 is what we expected our ORP values to be. So for our COD data, we see that COD has the ability to decrease in our treatment system. And this is our different ratios. We have our food processing wastewater, and 1 to 0, 0 to 1 municipal wastewater, which is our untreated. And they act as our controls. The 0 to 1 and the 1 to 0 ratios are our controls. And the food processing wastewater, municipal wastewater, is the actual wastewater that we sourced from these plants. And this is during the second stage. And we can see that in the first graph, COD goes up as high as 5,000 megagrams per liter, but it decreases in the second graph, which tells us that this system has the ability to reduce carbon. And we use that to calculate our enhanced carbon removal percentage. And we see that in some of the treatments, we have carbon removal as much as 50%, around 50%, in some of them 25 to 50%. So we did similar work for the nitrate concentration, and in some of the treatments, the nitrate concentration was too low to be detected. But we have removal around the same thing. We have our nitrates being removed. And once again, we have removal rate of around 50%, and sometimes it's even as high as 75%. And we also did ammonium nitrates. So in this system, we can also see that ammonium is also being removed during the treatment process. And during all of the stimulated wastewaters, whether we used it synthetically or real wastewater, ammonium still has the ability to be removed from the system. The same can be seen with TN. We see that TN is also lowered during the first phase, in the second phase, after the first phase, after aeration, during the treatment system. So what are our implications from this study? I would like to start off by saying this is a proof of concept study, and we just wanted to see how well this will do in actually being a method of tertiary treatment for wastewater remediation. And so far, we found that current results implicate that co-treating these wastewaters is indeed beneficial, as it significantly enhances the reduction of pollutants like carbon and nitrogen, more than we would expect by just diluting these wastewater products. And we're still analyzing data continuously until we satisfy the answer for our first question, which is which ratio works best. But so far, we can see that the preliminary results indicates that the 1-to-1 ratio, which is 50%, works best, as it lowers nitrates, lowers ammonium, lowers COD, has a great carbon removal percentage, and it also moderates pH, which tells us that this system treats our wastewater the most efficiently. And I would like to thank the North Carolina Water Resources Research Institution for funding this experiment, as well as the City of Greensboro and the North Carolina Food Innovation Lab for assisting us with our work. And I am available for questions if anyone have any questions. Oh, and I just want to mention, this is a photo that I took at the T.Z. Osborne facility, which is the aeration tank is located in about this area, which is where we sourced our water from.
Thank you, Jonaé. Any questions?
Hi, Jonaé. So I forget the exact terminology. Was it food process wastewater?
Food processing wastewater, yes.
Food processing wastewater. So I imagine that is going to vary a lot depending on what industry of food processing you're looking at.
Correct.
So how do you think that would impact like applying this to a different wastewater treatment plant, like broadening your application?
Sorry. Yeah. So like you mentioned, depending on the industry that produces the wastewater, the parameters of the wastewater is going to be a bit different, but the idea, the concept still works. Since we're removing carbon and nitrogen, the concept is still going to be the same across the board, but it's just going to be like different rates of carbon removal, different rates of nitrogen removal.
Hi, thank you for your presentation.
Thank you.
When you were measuring ammonium nitrogen, did you take into account the pH fluctuation to account for the species family of ammonia and ammonium?
So actually, that is why we recorded pH and ORP as supporting data to our experiment. We also want to go in and do a nitrogen balance in our experiment as well. I will say this, that the food processing wastewater that we got was water that was ready to be released into a watershed, whereas the municipal treated wastewater still had yet to go through its final treatment phases. So that also played a part in the levels of nitrates and nitrogen species in our wastewater. But yes, that's why we collect pH and ORP data, so that we can have supporting data for species.
Thank you.
No problem.
Any other questions?
Thank you for the presentation.
Thank you.
I have a just a small question. So when you said measure COD, nitrogen, did you measure some other nutrients such as phosphorus to see how those kind of nutrients in especially the food processing waste varies and affect the your system?
We don't do TP, no, but I will say we measure nitrates and ammonium using our IC system. So we have other anions and cations that are being measured as well, such as sulfates and calcium, lithium, sodium, stuff like that. But TP is something we do not measure, no.
Okay, got it. Thank you.
No problem.
Thank you, Jonaé.
No problem.
Now, back in concurrent session number 1, what I would do now is like him and haw for a few minutes so that we could get back caught up on time. But it's the last session of the day. So I think we're just going to keep rolling. How about that? Our next speaker is Emma Guertin, and she's going to talk to us about anaerobic co-digestion of alternate wastes. So we'll get you set up here and you'll be ready to go. Thank you, Emma.
Hi, everyone. Thank you for sticking around. I know it's the last session of the last day, so I appreciate y'all being here. My name is Emma. I'm going to be presenting on my master's thesis project, which is developing a robust anaerobic co-digestion process of food waste. And I work with Dr. Francis De La Reyes and Dr. Morton Barlaz at North Carolina State. So a key point in addressing increasing energy demands, growing populations, and trying to build climate change mitigation practices is working towards circular economy, where waste products are transformed into resources. One major waste resource here in the US that we can utilize for resource recovery and energy is food waste. USDA estimates that 30 to 40% of food waste is food is wasted in the US. And in 2018, that was 63 million tons of food being wasted. Where's all this food waste going? Majority of it is going straight to landfills, which we heard a little bit around from our other food waste people today. We have a major category going to landfills, another category going to combustion, as you can see in the yellow and the red on the pie chart. These two categories can have negative environmental impacts from the greenhouse gas emissions that come from this. This ends up taking about a quarter of the space in municipal landfills. This is the highest category of municipal waste in municipal landfills. So we're looking at ways that we can divert this waste from landfills and decrease greenhouse gas emissions. The way to do this is by zooming in on the other category of this chart that EPA puts together. These other categories include donation, animal feed, but the category we're focusing on optimizing is anaerobic co-digestion. That's what my project is dedicated towards, is how we increase the amount going to anaerobic digestion and divert the other two more or less environmentally friendly options. So it has already been found that anaerobic digestion is the most environmentally beneficial food waste management strategy by Levy's 2021 study, where they compared the global warming potential of multiple different ways of managing food waste. We have, as you can see in 80, oh, I have a, yeah, 80 over there on the end has the most negative GWP. This is because of the biogas that's produced from 80 that we can use to then as energy source, whether we use this for heating plants, that this process is being done at, or we can scrub and clean and outsource to grids and vehicle power. So the science is there that this is the most environmentally friendly way. And there are policies in the US that are starting to be spread statewide as California and Massachusetts that are banning food waste from going to landfills. So what is the issue? We haven't gotten there yet. There are a few knowledge gaps with food waste that have slowed the full-scale adaptation of these methods. Primarily, food waste is very variable in composition, depending on where you're getting it from. And you also don't have much control of what food waste you're getting at plants. It's whatever's locally near you, right? Grocery stores, restaurants, it can vary. The composition varies a lot. So this project is dedicated toward identifying the mixtures of food waste that result in optimum methane production. Optimizing methane production is profitable for plants that are doing this. As I just mentioned before, how this can be used to heat their plants or they can outsource it. So we're looking to find the food waste mixtures that result in optimum methane production. I know how I just said you can't control what food waste you're getting. So by understanding what the impact of different food waste types do to methane production, we can help kind of manipulate what time we're putting food waste in. For say, if a plant is getting high volumes of protein, what is that going to do to their plant that they've been feeding lipid this whole time? So I'm looking at specifically the food waste types of fruit and vegetable, looking at fat, oil, and grease, and we're looking at protein, which is meat waste. We're also trying to understand how the microbial communities respond to shifts in mixture types. As you've heard today, how important the microbial communities are to the anaerobic digestion process. They are the core of the process, and if we are upsetting the microbial communities or changing them, we won't get optimal performance. So this is a bit of a chart of the process of anaerobic digestion. starting with food waste, the organic substrate. So anaerobic digestion is a biological process of the breakdown of organic substrate into biogas, methane, and carbon dioxide. It's a 4-stage process with specific microbes at each stage to perform each job. It's sequential, like we've heard earlier today, that each one needs to be done properly before the next one. Correct. So we have hydrolysis, which is the breakdown of the complex polymers of carbohydrates, proteins, and lipids into more soluble, smaller monomers, which are then used in acetogenesis to produce VFAs, which are then used by acetogenesis bacteria to produce acetate, hydrogen, and carbon dioxide. And then at the end, methanogenesis that uses the acetate and hydrogen and carbon dioxide to produce biogas. So food waste can be used as a substrate in co-digestion. There have been multiple studies looking at monodigestion of food waste, but there are often process performance issues of just digesting food waste on its own due to low pH issues and lack of other nutrients that microbes need to perform these stages of anaerobic digestion. So using it as a co-substrate in other inoculums such as municipal solid waste, biosolids, and animal manure, we can kind of counterbalance those issues and supply the other nutrients that these microbes need. Mine specifically is looking at municipal solid waste. But yeah, co-digestion process, using food waste as a co-digestion in the process can help eliminate some of these issues. So my experiment followed a unique simplex centroid model design to look at the different mixtures of food waste types that we've been talking about. So I started with 18 reactors that were all loaded with synthetic municipal solid waste as the inoculum, and 6 starting each vertex A, B, and C. So for example, at A, the 6 reactors that started at A were only fed fruit and vegetable waste at the beginning at stage 1. At B, they were only fed fat, oil, and grease waste. And at C, they were only fed meat waste. After the methane production had peaked and began to decline, I then destructively sampled 1 reactor from each vertex to be used for microbial community analysis later on. The remaining reactors were then loaded towards mixture points D, E, and F. And these are 50% of 50% the original substrate type and 50% a new type. So for example, the reactors that had moved from A to D were fed 50% the original type, which was fruit and vegetable, and then introduced a 50% of fat. And the same goes for E and F. Again, once methane generation had peaked and began to decline, I then removed a reactor from each pathway and loaded the remaining reactors towards G. G is equal parts of all 3 types of the 3 substrate types. So this model lets us collect data across all the different mixture pathways and gives us the chance to study process performance data such as methane production and also microbial community analysis after we run community analysis on the microbes or on the reactor samples. And we can compare from how they looked like at mixture point A when they're just carbohydrates and how they looked at the end when they had gone through stages D and G to the ending point. This is the setup of my reactors. You can see I had 18 of them, as I mentioned, that were loaded with food waste and synthetic municipal solid waste. I also had 3 control at the end. They were connected to gas bags at the bottom here that collected the biogas that was produced, so I could measure the concentration, the composition of the gas, as well as calculate production rates. We also had leachate connection bags that collected the liquid to recirculate throughout the system every few days. So we'll first look at methane production rates in this chart here. So we have days on the x-axis here from when I started the experiment, and then production rates on the y-axis and milliliters of methane per day per gram of food waste. You can see pretty evidently in the green here, we have the meat waste reactors having the highest production rate of the 3 substrate types. We also can see that a little bit in the purple, which is the fat waste, a little bit of a lag phase here, where it took a little bit longer for those reactors to reach peak production rates, which is expected with the fatty waste of lipid waste. So in stage 2, this is when I loaded, stage 1 was when they were just 100% that type. So at stage 2, I then loaded the reactors towards those mixture points D and F, if that was, remember, the 50% new, 50% original combination. So at the red dotted line here is when I loaded the fruit and vegetable reactors towards their new mixture points in the blue peak. You can see it's similar to what it was in stage 1, suggesting that these microbes were able to handle the change in substrate, as well as lipid. Lipid had the similar performance to what it was in stage 1. No more lag phase, which is promising that these communities had been able to adapt faster, and similar production rates to they were in stage 1. But there is a drastic drop in the peak production rates from protein stage 1 to stage 2. This suggests that the microbes that have originally formed from the protein from the meat waste in stage 1 were not able to adapt to these new changes. We found similar results in stage 3, when represented by the yellow dotted line at the end, where all reactors were then loaded towards the combination of the 3 types. Again, similar performance from the fat and the fruit and vegetable waste reactors, and similar to stage 2, but much less in stage 1 for the meat waste reactors. So these results really highlight the importance of the pre-existing communities that are formed when you in anaerobic digestion. So in the meat waste reactors, we had a significant drop in peak production after they were introduced to these new substrate types. This suggests that starting off plants, they should not be loading in protein as their initial substrate, as these communities cannot handle changes and are not robust and resilient communities. And for the fog and for the fruit and vegetable reactors, these ones were able to handle the changes in substrate types, if not even slightly increasing as we went along, introducing these new substrate types to these communities. Another thing to mention is ammonia concerns. So commonly in food waste and protein specifically based reactors, there is a high concentration of ammonia in the reactors due to the high content of nitrogen in food waste. At the end of my experiment, I had about 4,500 milligrams of ammonia per liter in my reactors. A common threshold that was referred to in literature is McCarty's standard of 3,000 milligrams per liter from his study. And so that is kind of one of the cutoffs that people include as the ammonia threshold. But there is a wide range in literature of reactors being able to perform at much higher concentrations of ammonia. It really depends on the inoculum you're using, the substrate type you're using, operational conditions. So it's kind of a wide range, but it is possible that the reactors were impacted by the high ammonia concentration. Specifically, methanogens are susceptible to ammonia. So it's possible that this drop in methane production from the protein-based reactors was due to the ammonia inhibition compared to the other reactor types. But we will get better insight when I'm able to do microbial community analysis on the samples that I have collected. So this next stage is to run DNA sequencing on the samples I have to understand how the microbial communities themselves have actually changed instead of just basing off of the production data. Once we have the microbial communities, we can then put this with the production rates and see if this is actually attributable to the community changes. So the first goal from this is to find the specific communities that exist at the different substrate types and the types in the mixture types. And I'll also understand how these communities adapt and change to these substrate changes, so we can understand the resiliency and the robustness of varying communities to the types of food waste. In the end goal is to have optimal operational procedures of food waste loading to help plants know what to expect from loading food waste to their reactors. So if they are going to introduce protein to a new system, how is that going to impact their microbial community in the reactor, and what can they expect from gas production? And also we can kind of form a procedure on how to maintain and build resilient and robust microbial communities. So I am happy to take any questions. These are my references, but yeah, I'm happy to take any questions. Thank you. Be nice, Joni.
Good job, Emma.
Thank you. Thank you.
I have several questions.
Okay.
So the first one, when you added the substrates in it, what's the unit that you were using when you added substrate? Was it gram, wet, or COD?
I loaded it on COD basis. So I loaded around 40 grams of COD to the reactor. So it's a different volume of substrate, like a lot less lipid than it was cellulosic, so a lot more fruit vegetable than it was compared to the grease waste. But yeah.
Okay. I'm a little curious about your your next stage when you start mixing things. Do you add extra substrates like mixture this time to it, or are you adding just some, for instance, for the protein reactors, are you just adding some carbohydrates in there, or are you adding a mixture of meat waste and fruits and vegetable?
So you're asking how I mixed for stage 2 in stage 1? Yeah. So I loaded initially like that 40 grams of COD of, we'll say protein, the protein reactors. And then for stage 2, some of those reactors were then loaded towards, I'll go back to So some of the reactors were loaded from, so C is protein, some of them were loaded to E and F. So at E, those reactors that went towards E were fed 20 grams of COD, so half of the volume loading rate of protein. So there was getting some protein and some are getting, the rest of it was, that 50% was coming from the COD of fruit and vegetable.
Okay.
Does that answer your question? Or
Yes.
Okay.
Yes. So I guess expanding from this question, would there be concerns of overloading the reactor, the microbial communities in this case, if not all of the protein originally got degraded, would there be overloading problem and sudden drop of pH, those kind of problems?
I didn't see drop in pH. I did.
That's interesting.
Yeah, I might actually I had a weird pH issue this whole time. I've had a very high pH, upwards of 8 in my reactors throughout this whole experiment. From specifically in the protein reactors had a very high pH, and we think that's from the ammonia concentration. And the I think right now the production rates are our biggest storytelling data that we have right now until I can do stats analysis and the community analysis. But yeah, there was a second part to your question that I'm not answering. What was it that you asked?
I don't remember. Overloading?
Overloading. Yeah. So I think that is another thing I've been is that loading that protein in stage 2, because those reactors already had protein and the production rates have decreased. So there should be no substrate, the substrate should be much less and it shouldn't be overloading. But I think that the introduction of more protein could be a reason why this drop is so significant from stage 1 to stage 2 in the protein reactors.
Okay.
Yeah.
Thank you.
You're welcome.
Other questions for Emma?
Hi.
Hi.
Very briefly, you mentioned that you're in your future work that you're going to do gene analysis. Is that correct?
DNA analysis?
Yeah.
Yeah. Yep.
So were you thinking of doing something like amplicon sequencing with your 16S, or are you thinking of like metagenomics kind of approach?
Right now we're planning on doing 16S or RNA sequencing. It's kind of tricky with solid waste. I have a lot of plastic and a lot of weird things in my reactors. There's a long processing process to get to the sequencing, but we're planning on doing 16S or RNA sequencing. Yeah. I'm happy to hear your opinions on which one to use if you have one. But we just from what our lab has been doing, it's right now 16S.
Yeah, because I'm actually doing I'm looking at community makeup with cyanobacteria and Legmici, and we did both basically. We used primer for amplicon sequencing. We also did metagenomics. Depending on the software you use to identify stuff from 16S or other primers, it really depends on how good your existing database is. But with metagenomics, you can sometimes find things just right there. I mean, I feel like it's a better resolution, but that's my take. If you had the resource and the time, maybe you want to dive into both to see which approach might be more preferable.
Yeah, definitely. Thank you for the recommendation. I definitely will do that.
Other questions for Emma? No, we can. Thank you very much, Emma.
Of course. Thank you.
So I think we can move into kind of a general session. If there's questions that you want to ask for any of the speakers, yeah, I'd actually like to start, if that's okay, because I have a question for you all the presenters. I mean, the science is really interesting here, but I wonder if you could put on your future goggles for a minute and think about, what's the application? I mean, what is the application at the community scale or the individual business scale? I mean, what do you think is actually going to happen here that would be able to use the technology?
So like I mentioned quickly that there is already bans of food waste being put into landfills, specifically in California. Massachusetts is on their way. Seattle it's a local level of banning food waste. So there's becoming a need for an alternative source of where this food waste is going, and to help reduce the greenhouse gas emissions that are coming from landfills. There's a really high potential for plants to optimize their gas production and make more profit from this gas production, and also help, follow these state legislator of banning the food waste from going to landfills in general. So that's my answer. Yeah. Yeah, great question. So my project is focused on municipal solid waste. And this also can be applied to wastewater plants or solid waste plants. So that's what I'm talking about when I say plants. Thank you. Do you want to take a hint? Yeah. Do you have a question?
To answer your question, like I mentioned, it's extremely difficult and kind of costly to treat food processing wastewater. I'm not sure if this is on, you guys can hear me, but the process, because it's so costly and difficult to treat, there is a big potential for like, how do I say this? Corners to be cut in a way. So the water that gets that goes to these receiving bodies, like our watersheds, is still kind of high in pollutants and toxins, which is why we have such a big issue with nutrients in our water sources. So when it comes to co-treatment of food processing and municipal treated wastewater, two industries that release a huge amount of water into our watersheds. Like, if we can find a way to mitigate that environmental impact in a way that benefits both industries, then we can kind of get them to kind of work together to reduce their environmental impact on the community. I know you wanted to make a comment.
Oh, yes. I guess my one comment is about anaerobic. I know it was in the last session, but for future applications, anaerobic digestion is useful anywhere that you have really thick waste that you need to treat and you don't have a lot of resources. And a lot of times when you put together a really thick waste and you leave it out, it makes anaerobic digestion conditions anyway. So that methane will be produced and released into the atmosphere, like Emma was saying, that we can recover that methane and use it instead of letting it go into the atmosphere. You know, using it converts it to CO2, which is still a greenhouse gas, but a much less potent one.
I agree with all of those comments. I just wanted to ask something specifically for my own study. For instance, for the Hezhou study. So for like in some wastewater treatment plants nowadays, there are already applications like trying to draw out VFAs. There are acetogenic reactors out there rather than to methanogenic digesters. So some of the plants, they might be facing the issue of methanogenic adaptation and methanogens using up the VFAs. And the Hezhou strategy specifically is what we're trying to find. We're trying to find easy methods that we can heat up the reactor somehow, and then by doing that for a short period of time, it can last. The effects will be lasted for a like 60 days of period, which we think could be effective. But of course, there needs to be a lot of pilot scale study or even full scale study done after the lab scale study has been proven effective.
Anybody else in the audience have questions that they'd like to ask our speakers? Well, you know what that means? We get to leave early. Thanks everyone. This was a great session, and one of the reasons why it was great was everyone is fully engaged, and I really appreciated all the questions. I think that made it more interesting for everyone. I think we learned a little bit more through those interactions. So thank you for that very much. Thank you for joining us for the 2022 NCWR WRRI conference. I'm also with NCWRA, so they're one of our sponsors. We'd like to thank you for on behalf of them, our staff, the sponsors. Thank you for joining us. Look forward to seeing you next year and seeing how this continues. So thank you very much for joining us today. If you need professional development credits, I think the forms are at the front desk. All right. Thanks.